cuisine. In his book Life in the Rocky Mountains, Warren Angus Ferris recounts his adventures in the headwaters of the Missouri, Columbia, and Colorado rivers from 1830 to 1835. Back then, bison fed on diverse mixtures of plants, and Ferris' crew fed on bison, as indigenous peoples had done for eons. As Ferris notes, bison in poor flesh were the worst diet imaginable, but as they became fat, no other meat could compare. Quote, "With it, we require no seasoning. We boil, roast, or fry it as we please, and live upon it solely, without bread or vegetables of any kind. And what seems most singular, we never tire of or disrelish it, which would be the case with almost any other meat." End quote. His comments raise two intriguing questions. First, why didn't mountain men require any seasoning for bison meat? Second, how is it possible not to get sick and tired of eating the same food day after day? The phytochemical richness of bison diets provides answers to both questions. Phytochemicals season meat and further enhance the ability of meat to meet needs for a much wider range of nutrients than meat from bison in poor condition. Palatability and human health are linked with the diets of herbivores through phytochemicals in the diets of herbivores. That includes not only energy, protein, minerals, and vitamins, but also the tens of thousands of phytochemicals plants produce. This rich pool of compounds is increasingly recognized as responsible, as a complex whole, when trying to understand how plants promote health in herbivores and omnivorous humans who eat phytochemically and biochemically rich plants and meat. The benefits to people accrue as wild and domesticated herbivores assimilate some phytochemicals and convert others into metabolites that become part of muscle, fat, and milk. This rich tapestry of compounds promotes health. That is like, but distinct from, the benefits realized by eating phytochemically rich herbs, spices, vegetables, and fruits. This enlarged pool of compounds, phytochemicals and metabolites produced by animals from plants, should be considered when attempting to understand the benefits to humans, such as dampening oxidative stress and inflammation linked with cancer, cardiovascular disease, and metabolic syndrome. Phytochemical richness may be one reason why people have decidedly lower inflammatory responses after they eat the meat of kangaroos foraging on diverse mixtures of native plants than when they eat the meat of cattle feeding on high-grain diets in feedlots. The metabolic effects of eating meat from animals foraging on phytochemically rich diets are partially due to the ability of phytochemicals to reduce oxidative stress and curb inflammation. Low-grade systemic inflammation, characterized by elevated levels of cytokines, contributes to metabolic disease, type 2 diabetes, heart disease, cancer, and arthritis. Cytokines respond within a meal. Anytime we eat a meal, we experience inflammation, and we have an increasing likelihood of developing diseases when we eat a steady diet of ultra-processed plant and animal foods and sugar-laden foods and drinks that elevate inflammation. Moderating inflammation by eating wholesome foods can prevent or treat disease. Several phytochemicals in forage-finished meat and milk are in quantities comparable to those found in plant foods with anti-inflammatory, anti-carcinogenic, and cardioprotective effects. As meat and milk are often not considered sources of phytochemicals, their presence has remained largely underappreciated in discussion of nutritional differences between feedlot and pasture-finished meat and dairy, which have focused on omega-3 fatty acids and conjugated linoleic acid. Our studies of bison illustrate these points. Compared with bison finished on corn, meadow hay, and alfalfa hay in pens, meat from bison finished on phytochemically rich rangelands has higher amounts of compounds with benefits to bison and human health. Among many others, these include polyphenolic compounds, tocopherols, keratin, and omega-3 fatty acids. In contrast, less desirable compounds, such as advanced glycation end products, triglycerides, and short-chain azylcarnitines, are higher in pen-fed bison.